Now, for the start, I could tell you that these motor proteins are enzymes. So they hydrolyze ATP and use that chemical energy to produce the work that you saw in that last video. So, for example, M is for motor protein here. It binds to an ATP molecule, it hydrolyzes it, and then it releases the products in a sequential manner. And during this one ATPase cycle, the motor produces work and some kind of step along its cytoskeletal track. So for that to happen, the protein itself has to undergo some kind of structural changes during this enzymatic cycle that allows it to walk along the track. And now what I'd like to do is actually to take you to two animations that were made by Ron Milligan, Graham Johnson, and myself. And they really recapitulate decades of work by hundreds of investigators studying this problem. And this movie here shows the myosin motor and how it's thought to work. This is a myosin motor that might be found in your muscle. And the goal of this myosin is to bind to this actin track and slide the actin filament so that your sarcomere can contract and your muscle can contract. And I'll show you that at the very end of this lecture. But let's see now how it works. Here's the motor domain here. It binds to the track, it releases phosphate. Actin causes phosphate release, and that causes a big rotation, a part of that motor, a 10 nanometer displacement that is the part that causes the movement. Now ATP comes in, that kicks the motor off the actin filament, hydrolysis occurs and recocks the motor so now can bind to a new filament and undergo a stroke. Here it is again, phosphate release, the stroke, ATP binding, detachment, and recocking. And it's millions of these events that happen in your muscle, many of these cyclic binding, stroke, release events that result in the contraction of your muscle. Now, myosin is made to work in these big ensemble arrays with millions of other myosin molecules causing muscle contraction. But other motors in the cell have to do it solo. This is a kinesin motor here. And it may be attached way up here somewhere to its organelle cargo. And it's thought that even a single kinesin molecule, or maybe just a few kinesin molecules, are what are transporting organelles and mRNAs, proteins inside of cells. So the kinesin motor has to be very good, even acting solo, to move along continuously along a track without letting go. Very different from muscle myosin, which can detach for most of its cycle, just produce a brief stroke and then release. So let's see how this works. First of all, here's the motor domain. And now it binds to the microtubule. The microtubule kicks off ADP. ATP comes in here. And now look at this yellow element here. It zippers up along the side of the enzyme. And you'll see in the next one here, that zippering causes the rear head to be displaced from the rear binding site and to move to a forward binding site over here. It's this motion of what we call the neck linker here that pulls the rear head from this forward binding site. It moves around randomly, locks onto a new binding site. And in this way, the motor can work in what we call a hand-over-hand manner to move continuously along a track. And in fact, it can move for hundreds of ATPase cycles without letting go due to this coordinated action of the two heads. But how do we get to a model like this in the first place? Well, how do we collect data that even gets us close to thinking about a protein at this level of detail? So that's what I'd like to tell you about. How do you study the mechanism of a molecular motor? Well, there's several tools that we can use, very powerful tools that we're endowed with in modern biophysics right now. So one thing that we'd like to know is what the motor looks like. And we'd like to know this in incredible detail, at atomic resolution detail, so we know exactly where all of the amino acids and the side chains are positioned in this motor. And that's the job of X-ray crystallography. Here is a structure, X-ray crystal structure of the kinesin motor domain. And what you can see is the ATP in the active site. This is a ribbon diagram, and over here is a space filling model. And this was done by our lab in conjunction with Robert Flederic's lab at UCSF. So this is the detail that we really need to know to begin to understand protein structure and how it changes during this enzymatic cycle. But in the case of kinesin, it also gave an answer to a very old question in our field, which was, are kinesin and myosin at all related? And well, before the kinesin crystal structure, I think the answer that everyone thought was no. It made sense. You know, kinesin is a microtubule motor. Myosin is an actin-based motor. If you look at them, they're totally different in size. Kinesin is shown here in red, and this myosin motor domain is much bigger, as you can see here. And in addition, if you asked a computer to line up the sequences of these motors, the computer came back and said there's no sequence similarity between kinesin and myosin at all. Aha, well, surprise, we got the crystal structure of kinesin and looked at it in detail compared to myosin. And in fact, there was a remarkable structural similarity in the core part of these molecules surrounding the nucleotide. And this shows beta strands of kinesin and myosin in yellow and green. And you can see there's a remarkable overlap between the beta strands in these two motor proteins. In addition, all of the helices that you see here for kinesin overlap with similar helices in the myosin motor as well. This is no accident. This must indicate that the kinesin motor protein in myosin evolved probably very distantly in evolution from some kind of common ancestor. And that made a huge difference to the psychology of the field. People always thought, well, if I'm studying myosin, then, well, kinesin's another kind of motor that I don't really need to pay attention to, or vice versa. But this information indicated that the myosin field and the kinesin field were really studying variations on a common kind of motor that evolved very early in evolution. Not only that, but the structures of the motors also revealed a surprising similarity to a whole other class of proteins, which are the G proteins here, which people don't even think about as conventional motor proteins. But they also bind nucleotide, GTP, hydrolyze it to GDP, and they need to change their shape in order to act as switches in signal transduction cascades and other kinds of activities like that. And indeed, looking at the structures here in yellow, there was a common core element surrounding the nucleotide that was similar in these proteins. So we have some guess now about how all these machines evolved. Probably very distantly in evolution, there was a great invention, a nucleotide switch, something that learned to bind nucleotide, hydrolyze it, but not only just hydrolyze it, learned to change its shape when it went from 3 phosphates to 2 phosphates, like ATP to ADP or GTP to GDP. This gave rise to 2 different kinds of machines, the G protein family and some motor precursor that may still exist in biology or may have been lost, but probably was evolved very early in evolution. And this motor precursor then evolved 2 different branches. One motor that learned to walk on microtubules, another one that learned to walk on actin. And wow, this was such a great invention that evolution then produced many subclasses of these motors, a whole bunch of different kinesins, as I described to you, a whole bunch of different myosins. Now, in addition to getting some idea of the history of these motors, we also just learned a lot by comparing these structures to one another. And I don't have time to go into this in great detail, but what seems to be the most ancient and common theme that is shared between myosin and kinesin and even G proteins is how these proteins bind nucleotide, hydrolyze it, and then change their shape when the protein goes from an ATP to an ADP state or a GTP to a GDP state. So the nucleotide switching mechanism seems to be very similar in all these proteins, this core element here shown in blue. But onto this core switching part of the motor of the enzyme, nature has plugged in a bunch of different mechanical elements. Here's the myosin mechanical element, this big, huge lever arm that you saw rotate. And in kinesin, there's this much smaller little peptide that threw the partner head forward. So the mechanical elements have learned to work with this kind of common switch unit. And in fact, even in the myosin and kinesin families, there are a number of different variations on the types of mechanical elements and how they work.